Your sleep clock is the master switch for your DNA repair rate. Most people think of rest as passive recovery, but deep slow-wave sleep turns your body into an active molecular construction site during specific windows. When you enter that phase, waste products like amyloid peptides are not just sitting there. They are actively flushed out by a system called lymphatic fluid clearance. This precise timing allows enzymes like SIRT1 to optimally engage in epigenetic remodeling, essentially tightening or loosening the control dials on your genetic code. If your schedule is off sync with these endogenous rhythms, you are not resting. You are accumulating molecular debt that shortens biological lifespan far faster than aging alone predicts. The deeper cycles of sleep do more than clear cellular waste. They actively reset your body's entire internal stress calibration system. As you shift into those true restorative stages, the nervous system dominance shifts to parasympathetic control. This change dramatically lowers chronic inflammation by inhibiting key inflammatory signaling pathways like NF-kappa B. Furthermore, this endocrine downshift allows for a crucial drop in circulating cortisol levels. We know that consistent sleep architecture directly corresponds with improved heart rate variability metrics, a clear signal of heightened vagal tone and innate biological resilience. The systemic reset we observed in autonomic balance goes deeper than just calming the fight response. It involves optimizing your fundamental molecular programming at the genetic level. Think about it. Specific timing windows dictate when key enzymes, like methyltransferases and histone acetylases, achieve peak efficiency. These are not random repair agents. They are biological switches that tighten or loosen control dials on your DNA itself. When your sleep phase is out of sync with these endogenous rhythms, the machinery responsible for maintaining genomic stability slows down. This misalignment causes epigenetic drift, a subtle yet measurable acceleration toward metabolic dysfunction, far faster than simple calendar aging would suggest. The true cost of temporal misalignment is not just genetic drift. It is the degradation of your cellular power plants. The timing window for endogenous remodeling acts like a precise biological keyhole. During these optimal phases, essential enzymes, specifically methylation machinery and histone acetylases, do not merely repair damage. They achieve peak resonance with mitochondrial function. This means alignment is about optimizing enzyme performance itself. When that clock slips by even small margins, the efficiency of this molecular work drops precipitously. You are functionally operating your cells at half throttle while believing you are resting fully. The difference between slow decline and sustained vitality comes down to finding biological synchronization. So how do we objectively measure whether our internal clocks can actually be tuned? The failure is not just in missing optimal repair windows. It is deeper, embedded in your master timing mechanisms themselves. Your endogenous clock genes, the core regulators like BMAL1 and SLOC, are designed to run complex internal programs at precise times each day. When sleep architecture becomes inconsistent, those rhythms fail to entrain properly. They drift out of alignment with the solar cycle's demands for peak metabolic function. This molecular desynchronization is what accelerates epigenetic instability. It is not just a subtle slowdown in repair enzymes we saw earlier. It fundamentally breaks the body's inherent capacity for genomic stability on a systemic scale, creating an internal temporal chaos that affects every cell simultaneously. If your biological clock cannot maintain this tight synchronization with external cues, you are essentially putting your own life cycle out of sync with evolution itself. And the implications for predicting age-related functional decline based solely on blood work are becoming shockingly apparent. The crisis is not just metabolic debt or genomic drift alone. It is a far more subtle breakdown in molecular timing that dictates whether your cells can actually resonate with optimal function. We must understand alignment not as simply being in sleep, but about achieving peak biological resonance during specific windows of activity. Think of the cell machinery like an orchestra. If key components are tuned incorrectly, even slightly out of phase due to temporal misalignment, the entire system does not just play poorly, it fundamentally fails its core function. When these timing cues drift, methylation and histone remodeling do not proceed at a slower rate. They fail to achieve their necessary functional coupling with mitochondrial energetics. This means we are observing an intrinsic failure of cellular power transmission itself. The data reveals a fundamental shift in perspective we must embrace. It is not enough to simply maximize sleep duration. optimization demands precise temporal synchronization. To move beyond generic wellness, you need protocols that actively enforce the correct biological timing windows for epigenetic remodeling. Specifically, pairing consistent light-based phase shifting with objective heart rate variability measurements allows you to quantify your vagal tone and validate cellular resonance investment. This shifts the metric of success from merely logging hours slept toward achieving genuine autonomic coupling in rest. If you want the full research archive on these protocols, visit jmthomasofficial.com. The evidence does not just suggest better sleep is beneficial, it dictates a new standard of biological investment for longevity. Stop aiming merely for duration. Focus exclusively on precision timing architecture. The actionable takeaway here is this: use consistent morning light exposure paired with targeted evening thermal shifts to actively entrain your endogenous clock genes and enforce those optimal epigenetic remodeling windows. This moves you from general wellness into true molecular optimization, treating time as the most precious resource in biological maintenance. For the complete protocols supporting these chronobiological findings, visit jmthomasofficial.com.